Hello everyone, good afternoon. So as Matt said, it's a great pleasure to be here. I've been here before and spoke with Matt and other people, and it's very nice coming back again. And as Matt said, I'm a clinical lecturer funded by the NIHR, and I work here in Cambridge. Essentially, I split half of my time working as an old-age psychiatrist, seeing people with dementia, memory problems, or all other sort of psychiatric problems. And the rest of the time I do research in dementia, mainly Alzheimer's disease and Lewy body dementia. So today I'll talk to you a little bit about what is Alzheimer's disease, what is Lewy body dementia, and what we are doing at least a little bit here in Cambridge. And Shores will give you a very good follow-up on that. So I just put a funny infographic there about Alzheimer's disease from the NIH. But that's very informative for me as a psychiatrist because it pretty much has a lot of the things of what Alzheimer's is. It usually affects older people, women much more than men, about double the numbers in women than men. It's usually over 65, as I said, and people with Alzheimer's disease have memory problems, as we all know about it, but it's not just that. Alzheimer's disease is not just memory problems. What is more concerning, or what is really the problem, is that memory problems affect someone's day-to-day life. They stop them from doing their day-to-day things. They can't keep on top of any of their paperwork. They cannot attend appointments. They cannot manage their finances. They have problems with driving. Everything, all their life is really taken over by the disease. And that's really where the problem is, and that's what we really need to do something about it. And as you know, when we look at the pathology, you can see Alzheimer's disease in brain scans when it's at advanced stages. You can see it afterwards. Essentially, it affects all of the brain. The brain shrinks. You can see on the left a sort of normal brain, and you can see on the right how a brain with Alzheimer's disease is. In early stages, some areas of the brain are affected more, but as the disease progresses, it affects pretty much the whole brain, but also the rest of the body as well. And then a little bit on the causes of Alzheimer's disease. Probably you've all heard of the amyloid hypothesis and what really causes Alzheimer's disease. We have a lot of environmental factors that we try to identify, and we know a lot of them, mainly around cardiovascular risks that are involved in the disease. We have genetic variations, have early onset forms of the disease, which is usually familial mutations on some of the genes that are involved in these processes. We also have the apolipoprotein E4 gene that is for late onset Alzheimer's disease. It increases our risk of getting it about 4 times. We have both copies. And then we know that this interaction between genetic and environmental factors, we don't know exactly how, there is a lot of research, do cause the pathology that we see in the brain. We see amyloid plaques that are around cells. They are not inside the cells, but they probably produced inside the cell. We have the oligomeric forms and all of sort of other forms of amyloid. And then the cell secretes them and become extracellular deposit of the plaques. But we also have the tangles, the tau tangles, and Sorzo is going to speak more about that. I presume that come, usually it's the combination. It's not very clear what comes first or what is really most important, but both of them are really the hallmarks of Alzheimer's disease. And they seem to lead to loss of neurons in the cell that causes the shrinkage. But also we have neurotransmitter deficits. The synapses are lost in Alzheimer's disease, and that's what really causes the clinical symptoms. And then there's a lot of research, a lot of animal models, a lot of trials targeting both amyloid and tau, and unfortunately we don't have a disease-modifying treatment today. We don't have anything. We have things that can stop the plaques from forming or can clear them up. We have some compounds that may modify tau, but nothing really works in clinical practice. And that's quite disappointing, really, because in animal models that carry mutations, all of that, we can really treat them. We can make mice better, but we cannot make humans better yet. We have some symptomatic treatments in clinic that we prescribe, mainly acetylcholinesterase inhibitors to increase the levels of acetylcholine in the brain when someone is diagnosed with Alzheimer's disease. They have some benefits, but they are far from what we really want. And the combination of all of this is what we call Alzheimer's disease, and that's pretty much what's happening. Probably see a lot research nowadays is shifting from traditional amyloid and tangles. People try to look for other things treat, better ways to understand the disease. And inflammation is a lot in the mix now. There is a lot of new genetic variants like TREM2 coming into the play of how important they are, the role of microglia in the brain and how this might be important in Alzheimer's disease, and obviously how that might be involved in the whole disease process. It's still ongoing work, but perhaps if we find something, it might be a new target to modify or something to really do more about it. And that's what Alzheimer's is. It's the most common type of dementia. About 6 to 7 out of 10 people with dementia will have Alzheimer's disease as we know it. But what we know increasingly more and more, and what we do a lot in Cambridge, is to look at other types of dementia as well. Lewy body dementia is the second most common type of dementia in older people, and it's slightly different from Alzheimer's disease. The main hallmark of it is Lewy bodies forming within the neurons of certain brain regions, and then they spread. Similar pathology to what we see in Parkinson's disease, but much more broad in the brain. And it's actually very often misdiagnosed as Alzheimer's disease because of the way people initially present. But it has some different characteristics, both clinically, but also neuropathologically. People with Lewy body dementia often have Parkinsonism. As a result of that, they fall more often, they have more complications, so their disease progresses more quickly. They can often also have a lot of visual hallucinations, sleep disorder, REM sleep disorder. They have fluctuations in their cognitive function. They sleep a lot during the day. They might be awake at night, or they have a lot of unusual, unresponsive episodes. And they have slightly different cognitive profiles when it comes to cognitive testing. Some other areas are affected, and then they have less atrophy in their brain. And I'm listing all of those things because we now know all of these things. We can identify a different type of dementia because of a lot of research, and a lot of this actually started in the UK. People started seeing that people with dementia had slightly different symptoms and were able to define the different symptomatology. And we know that when we see neuropathologically, it's the second most common type of pathology, and often people clinically have a diagnosis of Alzheimer's disease, and when they have a post-mortem examination, actually it comes up that they have Lewy body dementia. And obviously, the clinical aspects of the disease are what we deal a lot with. The numbers are rising, as Matt said. I just put some of the statistics from the AER UK website. They have a very nice hub where they summarize a lot of statistics around numbers, financial costs, but they have even created a heat map of which areas in the UK are more affected, and that can aid a lot of aspects of research. The numbers are expected to double. I think Matt quoted a number of 500,000. Some other sources say 800,000 people nowadays in the UK. There are different ways of measuring that, but it is globally an epidemic, and is a big challenge because with the aging of the population, numbers are rising, and we really have to do something about it. And again, I put some more numbers there. The impact of Alzheimer's disease and other dementias is really huge. It's not just having memory problems, being at home and needing carers or people coming to support you. It has a huge impact on families, on the people themselves, on health costs, social care costs. People with Alzheimer's disease often have a lot of comorbidities. If you ever in Addenbrooke's, probably you all have families. But a lot of the people in Addenbrooke's and in every hospital have Alzheimer's disease. They're inpatients. They're very difficult to treat or support. And it's really something we really need to do more about, more research, and we need everyone involved. So here in Cambridge, there's a very big biomedical research center. I'll come to that. The strategy is obviously to try and do something about all types of dementia. We have clinical cohorts that are recruited to trials. We try to do proper assessments, very detailed ones, and then get them to trials. I'll speak a little bit about some ongoing trials that we have. Try to develop biomarkers, but also have the Cambridge Brain Bank, one of the biggest in the UK, where we try to get people to donate their brains and then use them for research purposes afterwards. So this is a map of the Cambridge Biomedical campus. It's essentially a world-leading comprehensive center. It has pretty much everything, very extensive neuroimaging facilities, all of the latest state-of-the-art brain scanners. We have now a 7T MRI Tesla scanner, 3T scanners, PET scanners, both PET-MR, PET-CT scanners, all sorts of world-leading neuroimaging studies can be done here. We have memory clinics on the same site, inpatient facilities, obviously, but there are also labs, the Fangist Center for Brain Repair, the Alzheimer Research UK Drug Discovery Institute, the Dementia Research Institute, the LMB. Everything is really on the same site. We now have industry, Abcam, and other companies coming in. So it's really the place to be to do cutting-edge research and come all together, really. Apart from the Biomedical campus here, there is also the Fulbourn Hospital. It's a psychiatric hospital, and there is a very well-organized research unit there, the Windsor Research Unit, which is essentially leading most of the local trials in Alzheimer's disease and other dementias. In 2007, there were 30 ongoing clinical trials, and we had about 370 patients into trials this year, Alzheimer and other dementias, either pharmaceutical trials or imaging or all sorts of other things going on. It doesn't look very nice on the outside, but it's very well organized and has everything that you require for a modern clinical research facility. One of the studies I'd like to mention today is called the Prevent Dementia study. It's a study that we are involved in Cambridge. We have 5 centers across the UK, and we have actually recruited 700 people to date, aged from 40 to 59. The reason for that is there is a lot of talk why trials fail, why we really need to do something about that. But maybe I should go. Yeah. I'll come on this side. Maybe it's better. But it's the other one now.
Can you move it closer to your mouth?
Oh.
That's what Ash told me anyway.
Okay, thank you. So yeah, we have 700 people recruited in the Prevent study. It's looking at people who, half of them have a family history of Alzheimer's disease or other type of dementia, and the other half not. The reason is we know that Alzheimer's disease starts way beyond any symptoms start. There is studies showing that amyloid can build up in the brain many, many years before anyone develops any symptoms. So we try to understand really people before they have any symptoms, if there is something that we can really pick up. They have very extensive neuropsychological and clinical assessments. They have brain scans. They also have all sorts of blood tests, and some of them consent to have CSF as well. And we are in the process of now doing the follow-ups. We have seen everyone at the baseline stage, and now seeing people at 2 years follow-up. There are some studies that are going to be published very soon around microstructure changes in their brain. We can already pick up things changing in brains of people using conventional MRIs if we look in detail to them and find out more. Another study that we're in the process of completing nowadays is the Nimrod study. It's called neuroinflammation in memory and other imaging neuroinflammation in memory and other disorders. Published quite a few papers on that. John O'Brien and James Rowe are the PIs, essentially looking at how inflammation might play a role in Alzheimer's disease, mild cognitive impairment, Lewy body dementia, frontotemporal dementia. So try to look at cohorts with all sorts of type of dementia and comparing them to understand a little bit more. People can have PET scans, MRI scans, blood tests, and CSF fluids, and we are in the process of analyzing a lot of that. And we used a fairly novel compound looking at neuroinflammation in the brain, and found that people with Alzheimer's disease and Lewy body disorders have different patterns of neuroinflammation, which helps us understand them a little bit better really at the clinical stage. My particular interest, and probably why I'm here, is my background is in neuroscience. I'm interested in epigenetics, and I know Abcam is one of the leading companies. I've bought a lot of antibodies before, and it's great being here. My particular work is understanding a bit better epigenetic mechanisms and how they are involved in dementia, how they might play a role in that. Why epigenetics is essentially because they are the mechanisms that have input both from genetics. Genetic variation can affect our epigenome, but also they are affected or influenced by environmental factors, both in the microcellular environment, but also external factors. And then is a way of measuring the interactions between the genetics and environmental factors. And we are studying that. There is a lot of work done in Alzheimer's disease predominantly, and try to set up some studies now in Lewy body dementia as well. And then I'm particularly interested in DNA methylation and sequencing that. I'll show you a little bit more afterwards. But there is a lot of work going on, and we collaborate with a lot of people. And I just put here, we did a review recently. There is a lot of genetic information known about Lewy body dementia. We know its heritability is around 35% at the moment, but there is still a lot to be discovered, especially in the epigenetics and also the interface of that, and that's what we are working on at the moment. Obviously, in Cambridge, we have a lot of cohorts. We also try to understand a little bit more the disease processes and the role of inflammation using the cohorts we have. From the Nimrod cohort and other cohorts that I mentioned, we have a lot of blood samples, we have CSF fluids, and try to understand a little bit more how the immune system is playing a role on that, how immune changes interact with genetic factors, environmental factors. We have sequencing data, DNA methylation data from peripheral blood, and immunoprofiling, which we do locally, and try to see how this relates with some brain measures that we have, and PET scans. And obviously, studying dementia but not studying the brain is not enough. We also do a lot of work in brain tissues. One of the issues with most of the dementias is that not all cells are affected in the brain. Some neurons get Lewy bodies or some neurons get tangles, but others not. And we don't really know why. And we try to study that, looking at specific neurons, isolating single neurons, and studying them instead of just getting bulk tissues, which cannot be very informative sometimes. We've set up a pipeline to do laser microdissection here in Cambridge. We have all of the equipment and all of the sequencing facilities available. So we get sections from people with Lewy body dementia, stain them using ABCAM antibodies for alpha-synuclein, and then isolate cells, and then do sequencing on them for DNA methylation profiling. And I've just put a video here of how the whole process works. There it goes, it works. So you see here a frozen section, and we've talked about the challenges of using frozen human material when it comes to immunohistochemistry before, getting neurons with Lewy bodies, neurons without Lewy bodies in separate tubes, and then sequencing their methylome. That's what I'm doing at the moment, but there are a lot of other opportunities as well. And then how this can help us understand the disease more. So that's the sort of things I wanted to talk about scientifically, but to make a difference, to make a change in dementia, we need everyone really. We need funding, we need industry, we need clinical research, we need basic research, and we need all of you really, and everyone's families. Recruiting people into trials, recruiting people to studies is one of the biggest challenges we have everywhere, not just in Cambridge. In Cambridge, we are very lucky because people are very aware of what's happening. So if you are interested in any sort of research or participating in any trials, or you have any families who are affected by Alzheimer's or any sort of other dementia problems, please visit this website. It's a national database from the NIHR to get people into trials. If we have something, people who are into trials will be the first ones to have the opportunity of doing that. There are all sorts of studies going on, neuroimaging, blood samples, just clinical assessments, looking at all sorts of things. So please, if you have any time, it's really important to have as many people as we can. We have the amazing NHS that can sort of get people into having all of the diagnosis and all of the clinical information. Linking that with research is very important, really. So thank you very much. I just need to mention some people who are supporting me. John O'Brien is our professor at the Old Age Psychiatry Group. He's running most of the trials that I mentioned. We work very closely with the clinical neurosciences here in Cambridge, Patrick Chinnery, James Rowe, and the people working with them. But also in Cambridge, we have the option of collaborating with a lot of people a lot of core facilities, the cell phenotyping hub, the sequencing facilities. We all work together to try and use everything we have available. And obviously there are people in other parts of the country that we work together as well, and internationally. And also the funders, very important. AR UK is really our most important funder, but NIHR and other institutes that we get support from. So thank you very much for everything.
So firstly, thank you to Leo for a fantastic overview to kick off our meeting. And I think, you know, in a short space of time, 25 minutes, a huge amount was covered there. But I think more importantly, it's a huge amount of information which is very relevant to the work that we do. And obviously a big focus there on two of our main research areas, lots of neuroscience, a huge amount of epigenetics as well. But I think looking at how much is going on in our campus just around us here, there are so many customers and so many projects and things that we can get involved in that can really add value to accelerating research. So again, back to the challenge set at the start of the meeting by both myself and Kim, really sort of look around individually and collectively to see, you know, all the things we hear today, what can we get involved in? Which areas of our skill set can really be applied to linking together all these different sources and insights and different projects that Leo summarised very, very concisely there. So does anyone have any questions? We've got a few minutes, but it'd be good to ask a few questions before we move on to the next slot. If anyone's thought of anything? Please.
Just a very clinical question, actually. You showed that we see now these differences, forms of Alzheimer's. Does this already connect to different treatments?
Well, at the moment we don't have any treatments that work, but if people have different diseases, they will need different treatments, so it's important to get it right. And misdiagnosis of Alzheimer's might be one of the reasons why trials have failed. Nowadays, trials use biomarkers for studies, amyloid scans or CSF fluid biomarkers. And we've seen in recent studies that we had, and that was a sort of global thing, that about one third of people that we thought had Alzheimer's didn't have it and have something else really. So that might skew numbers when it comes to trial results. So it's important to get it right and understand it better.
It's kind of linked to that. I was interested in the PREVENT study that you talked about, and you mentioned that was for dementia. So is that for all dementias, or like, are you going to? Is the idea that the study then segments out these different groups with different types?
At the moment, we try to get as many people with a family history of Alzheimer's disease. Obviously, not everyone will know exactly what the diagnosis is, or we don't know what exactly the diagnosis was, other than what the relative tells us. We don't see the people with the disease, only their families. But we try to get to focus on Alzheimer's for this particular study.
And which other key pillars of treatment do you see emerging, future areas of clinical research? So obviously tau and amyloid is going to remain, but they're both very important areas, but which other things do you see emerging moving forward?
I mean, inflammation is something that there is a lot of research now on the role of microglia, genetic susceptibility, how we can modify that, and we can modify inflammation, so there might be something there. A lot of other aspects of research, the infections and all of that, it's very difficult to predict really what will work. There are a few hundred trials going on already, but mind you, from a discovery in the lab to that translate into a clinical trial, that might take at least 10 years to sort of go through all of the steps. So it's difficult to know, really. Yeah.
Any more questions? No? Thank you very much, Lee.
Thank you.
Thank you.